Okay, thank you very much for that introduction, and thank you to the DAE and organizers. It's an honor to be invited to speak here today. Today I'll be talking about my work using stable isotope analysis of animal teeth and bones to reconstruct past climate change. As we know now, climate change is the world's most pressing problem. Just days ago, the UN warned of impending total societal collapse as a result of the deterioration of ecological systems caused by human action. Much of what we know about climate change, both present and future, we owe to direct observation and complex modeling of these data. But in order to gain a holistic perspective and a better idea of the full range of intensity, duration, and human scale impacts of climate change in our modern world, we must also consider the long-term data beyond direct observations in the historical record and turn to the archaeological record as well. So in this talk, I'll discuss the application of stable isotope analysis of archaeological remains to documenting climate change and shifts in past environments. But first, it's important to establish how stable isotopes in the modern Earth system can be directly evaluated in water and living plants and animals, and how relationships between isotopic ratios and climate variables can be established. Stable isotopes are naturally occurring and are non-radioactive variants of atoms with different weights. Different elements are useful for reconstructing different aspects of past climate and environmental variation. Today, I will discuss oxygen, carbon, and nitrogen stable isotopes. Measurements of stable isotopes are made with reference to an international standard. And for those of you not familiar, it's expressed in parts per mil, and the ratio is annotated as delta. So that's the terminology that I'll be using in this talk. And there are some guiding principles in understanding the range of variation possible within stable isotope data. So for example, oxygen has 3 isotopes depending on the number of neutrons. There's oxygen 16, oxygen 17, and oxygen 18. Measuring their ratio to one another provides an indication of precipitation amount, the moisture source, and also ambient temperature, among other variables. Using modern data sets, we can understand this variation both empirically and theoretically. In the top figure, we can see the global distribution of delta 18O in leaf water of plants. More positive values reflect higher temperatures and greater aridity and are shown as orange and red. More negative values are indicative of lower temperatures and greater rainfall amount and are shown in blue. Carbon, which has the stable forms of 12C and 13C, reflects photosynthetic pathways of plants, which vary based on climate conditions. So in this bottom figure, you can see predicted delta 13C values based on vegetative cover in Africa. Darker green areas like the Congo rainforest, where we would expect greater moisture and more C3 plants, would have more negative carbon 13 values. And the blue areas on the map, like those covering the Kalahari, for example, have more C4 plants and would therefore have more positive 13C ratios, reflecting the aridity. So we would expect to see this reflected archaeologically as well, these same patterns enduring through time and space. If we want to understand isotopic variation in the past, and therefore the climate factors affecting that isotopic variation, we need to turn to ancient faunal and botanical remains as a proxy. Though I also work on plant material, I'll today just be focusing on faunal data. So isotopic ratios reflect diet. You are what you eat and drink, essentially. So when we look at animal remains, this is another layer to take into consideration when interpreting the data. But what you eat is all around you. It reflects the environment. So these are still useful proxy data if we handle them correctly. When we have preserved skeletal material, we make choices about what to analyze. First of all, whether to include humans and faunal remains, whether to look at longer term sources like a femur bone that's going to reflect several years of formation, early life information such as that contained in a tooth, particularly the first molar that develops sort of during infancy and early childhood, and the last few years before death, something like a rib, which contains only maybe a year's worth of information. Or we can assess multiple different elements in order to get an idea of individual life history. In the case of bone, then, we are looking at time average data on the order of years. And when it comes to teeth, we can actually do sequential analysis, which can offer insight into subannual resolution, since teeth grow in layers, much like tree rings or seashells, or indeed, as sediment cores form. It's the same principle. So on the left here, I have an example of a demineralized bone collagen specimen. And from bone collagen, we can analyze the ratios of carbon and nitrogen and gain insight into herbivore diet, and by extension, the type of vegetative cover, whether it's C3 or C4, and by extension, then aridity levels. In tooth enamel, we can evaluate carbon ratios as well as oxygen levels, which, as I noted in the previous slide, is influenced by temperature and rainfall amount. And again, we can do this either sequentially or look at an average over time. So just like the spatially determined variation in stable isotope ratios I showed on the first slide, climate change through time and the resulting environmental, geochemical, and physical effects of that climate change will influence the overall diversity of stable isotope values in a given sample. So here I'd like to move on to talk about two different areas of my own research, focusing on the early Holocene and the modern era. I will talk about the nature of the data interpretation on different temporal and spatial scales, and why this matters for the application of stable isotope data from archaeological sources for the reconstruction of past climate change. So the first of these focuses on ancient Anatolia in the early Holocene. It covers a somewhat intermediate time span of about 2,000 years. So depending on what your research area is, this can seem like either a short amount of time or a very long one. And it covers a broad geographic region, about 200,000 square kilometers. The second study focuses on the more recent, late Holocene, we could say, modern London, and it covers an extremely brief time span of 10 years, as well as an extremely constrained geography of about 6 square kilometers, which is useful for the kind of research question in this case, where I'm trying to assess the direct relationship between climate variables and the isotopic signatures in the teeth. So in the first case, I've been working at the site of Uluru on the Aegean island of Gökçeada since 2015. I don't know that you can see my cursor, but the island is over here. And this site is important because it's the first Neolithic settlement in the Eastern Aegean, and people appear to have brought domesticate fauna with them when they settled the island in the early Holocene. This includes faunal remains, as I have pictured here, from things like sheep and goat and pig and cow predominantly. In the context of the process of Neolithization of Anatolia and the subsequent spread of farming into Europe, as I have here on the upper right picture, different zones of domestication and the spread of the Neolithic, the role of climate and environmental conditions is a large area of research. In this case, I sampled bone collagen as well as tooth enamel from sheep and goat, predominantly, at the site of Uluru to better understand the Neolithization process and climate context at the site as agriculture spread from these earlier zones, so zone A, C, into zone B. What we have pictured here is Western Anatolia and then up into the Balkans. And of particular interest is, the sort of pathway that the fauna are taking, whether it's an overland route up through, sort of mainland Anatolia, or if they're coming around the coast, the West Coast, and sort of island hopping up into the Balkans. And of course, now we know that it's, both of these processes are happening, but the interest is in the sort of nuance of the pathways that people took. And in particular, I was interested in whether there might be indications that fauna from the earliest levels at this site looked isotopically similar to fauna from the heart of Anatolia, from central Anatolia, or whether they had a unique isotopic signature looking like they were more from the coast and sort of just moving across into the island. So for reference, modern Turkish climate conditions vary between the western coast and the interior, where we have the major Neolithic site of Çatalhöyük, for instance. If the fauna in the earliest context at Uluru were originating from areas on the west coast of Turkey, so that's pictured here on the left side of the slide, I would expect that they would have lower carbon and nitrogen values due to increased humidity and a higher occurrence of C3 plants. And tooth enamels values would be expected to have a damped seasonal oxygen value, more negative values in winter months due to increased precipitation and decreased temperatures, as you can see here from these graphs showing precipitation and temperature for this region. In contrast, if they originated from the heart of Anatolia, I would expect higher carbon and nitrogen values due to increased aridity and higher occurrence of C4 plants. And in tooth enamel, I would expect more pronounced seasonal oxygen variation due to a larger range in temperature in this region, as well as overall more positive values due to lower annual rainfall amounts. So what I found was that, first of all, samples that originated from earlier levels at Uluru, as are pictured here as these green triangles, did seem to be more like the values of fauna from other sites on the west coast of Turkey. So here I have in the gray and the purple values from sheep and goat teeth from the sites of Yenikapı, which is outside Istanbul, modern Istanbul, and Ulucak, which is right on the west coast, actually not far as the crow flies from Uluru. And so these were more similar to each other than those from central Anatolia. And later fauna from the site of Uluru, which were presumably born and raised on the island in the later period, reflected values that I would expect for a very specific island climate context and environmental setting. And when I compared this to other contemporaneous levels at these sites, so sort of evaluating the overlap and standard deviations of these data here, there was spatial diversity that was stronger and more significant than any temporal trends in the data. And this data I would also just mention here from Yenikapı, Uluru, and Ulucak, I generated myself. These Çatalhöyük data are from previous work by Liz Henton and Jess Pearson. It's been some published and some not. And so there's clearly, I think here, looking at the means and standard deviations, a spatially determined diversity in these assemblages, even though they range over a 2,000-year time period, which is not an insignificant amount of time, and overlaps with events, climate events, like the 8.2 kiloyear event. And Çatalhöyük actually has the most variation, I think, because there is this larger temporal spread at this site, more so than these others. But I would argue that the sort of amount of variation that we see in the patterning in the data is more dominated by these spatial variants than the temporal ones. So this led me to more recently to assemble published faunal isotope data from bone collagen for this period, extending throughout Anatolia and the Levant. And I would also mention that's, more and more in the last several years, people have been publishing isotope data from fauna, but it's still relatively limited in the greater scope of things. I think we're seeing now sort of an explosion of this data. And these graphs on top here show carbon and nitrogen ratios. And we included all available taxa from multiple sites and found that there was a definite spatial variability even across taxa. So we're currently working on modeling this into a fauna-based ancient isoscape, much like those isoscapes I showed on the first slide for the region, which we can compare with other modeled isoscapes based on precipitation and vegetation, which we'll hopefully be submitting for publication soon. But I sort of have this Google Maps version of our map here showing the sort of significant spatial differences in expected isotope ratios. And so, for example, coastal sites, as I have here on the top left, tend to have overall means of around 6 per mil for delta 15 nitrogen and minus 20 per mil for delta 13 carbon. Whereas inland sites, as shown here by this star, tend to have an average of about 2 per mil more, so about 8 per mil for nitrogen, and minus about 18.5 per mil for carbon. So these are significant differences across taxa and across time. So keeping in mind the kind of isoscapes that I showed earlier, we hope that these kind of syntheses can be used to help better interpret spatial and temporal variation in archaeological isotope data going forward, and generate complex archaeoisoscapes that can be interpolated through time to better understand both the spatial and temporal dimensions of variation. So that brings me to my second case study, which is my work using a modern assemblage in the UK to better assess the relationship between isotopic values and climate factors themselves. So we can assess the archaeological data on its own terms, of course, and try to understand this range of variability. But we really need to, in order to make robust interpretations, have a good handle on, what is the primary influencing factor? Is it vegetation? Is it rainfall amount? Is it rainfall source, or is it the temperature? And how do these interact? So this project focuses on red deer, called elk in North America. The species is Cervus elaphus. And these deer were collected from Richmond Park, which is located outside London. And so I have here a historic map and sort of origins of this park as a, being for royalty to go hunt game. It was actually well outside London, but today serves as, with the growth of the city, is actually a park in the interior of London. And Donald and Norma Chapman were a husband and wife deer biologist team. And they tagged over 200 red deer calves within a day or so of birth. And many were the subject of a study on aging based on tooth eruption and wear. So we have extremely good resolution on the lives and dental formation of these individuals in a very constrained geographic area and temporal span. And these were actually stored in her garage attic, which I have pictured here, my sampling location about 10 years ago. So in this case, we have good data from the Metropolitan Weather Service located about 12 miles away from the park. So it's the top left graph here. And we also have measured delta 18 oxygen values from a global networks of isotopes and precipitation, GNIP, monitoring station that though it postdates the time period of interest, so these deer were collected, I should have said, between 1965 and 1975. They only started monitoring the isotopes and precipitation in 1980, but that goes through 2011. So that's this graph here. I think it still offers us, very good insight into the long-term trends in isotopic fluctuation in rainfall there. And these modern records of rainfall and temperature were used to compare to measured isotopic values in teeth, as well as converted temperature in this sample. And we found a good relationship between rainfall amount and oxygen values, but we're also able to evaluate the relationship between reconstructed temperature and rainfall amount. So here we have pictured rainfall amount graphed against the recorded temperature, as well as the black line, the calculated temperatures that we calculated using existing conversion equations in order to convert from the measured isotope values in the deer teeth to what the expected temperatures would be, which, as you can imagine, is a very useful tool for reconstructing paleoclimate, but how much variability is assessed by the rainfall, influenced by the rainfall amount versus ambient temperature. And so that we found that teeth were a good temperature proxy depending on the amount of rainfall. And so as rainfall increases by 1 millimeter, the difference between actual temperature as recorded by the weather station and the calculated temperature generated using existing conversion equations increases by about 1/10 of a degree Celsius. So your rainfall amount is really going to influence your paleoclimate interpretation. So these kinds of modern studies are therefore exceptionally useful for understanding what we mean when we talk about that large scale variation in isotopic values across time and space. But they also remind us that, like other proxies, the data need to be evaluated where they stand, and that the nature of the data is such that we are talking about averages of averages of averages. So here in these graphs, you can see that the actual variation in temperatures on the left side here was greater than the converted values from tooth enamel, if we were for instance, didn't know the ambient temperature in London during this time period and were just relying on the teeth to tell us, even though the actual measured isotopic ratios in each tooth displayed relatively high ranges of variation within each individual. So pictured here on the right, we have a difference of over 4 per mil, but it's not necessarily translating when we interpret the data. So just to sum up here, I would like to summarize and conclude, as I hope it's apparent from these 2 case studies. We need to incorporate stable isotope data from animal bones and teeth as valuable climate proxies when we consider past climate change on both spatial and temporal scales, but we need to assess them rigorously and also compare site-level case studies with large data sets, such as pollen from sediment cores and speleothem data. And so pictured here, I have a screenshot of North American data in the Global Neotoma Paleoecology Database, which includes the recently established faunal isotope database, which I'm part of. And I would just like to summarize and on the end on the point that compiling the data from multiple sources and ensuring the integrity of that data is really the next step forward in reconstructing past climate change and understanding its relevance for our current situation and future climate change. So thank you very much to the organizers and to the funders and to the audience for being here. Thank you.